Okay, welcome. This is Dr. Lindner, and we are going to be talking about muscle physiology. And then we're getting into a topic referred to as the NMJ, the neuromuscular junction. So, muscle cells are stimulated to contract. Remember, the term contraction means to generate tension. And muscle cells are stimulated to contract by somatic motor neurons. Motor neurons are the type of neurons that will attach to muscles, to the end plate of the muscle. And again, they're voluntarily controlled. The neurons make contact at the NMJ or the neuromuscular junction. So in this picture here is a nerve attaching to a muscle. This is the axon, one of the axon collaterals, which really is just a fancy way of saying it's a branch off the nerve. Okay, so it's a branch, it's an axon collateral. The axons are myelinated, so this is myelin. This is a Schwann cell that produced this fatty cholesterol-like protection to the axon. And the axon is going to make contact to the muscle at this level called the motor end plate. Now, the axon has branches coming off of it, right? Those collaterals, but at the very, very end, they are called synaptic end bulbs. Synaptic end bulbs. This right here is a synaptic end bulb. This is a synaptic end bulb. Right here is a synaptic end bulb. And why are these synaptic end bulbs important? These synaptic end bulbs are important because they house protein. What type of protein? Neurotransmitters. Neurotransmitters. That is the chemical messenger here when we talk about neurology. The very important neurotransmitter that is involved at the neuromuscular junction, and it is always excitatory, is called acetylcholine. Acetylcholine, which is abbreviated ACH. ACH is acetylcholine. So now what we're doing is we're taking this structure here where the axon meets the muscle, and we're looking at it here to the right. Again, here's the axon. The nerve impulses are traveling this way to the very, very end of the axon, and the end of that axon is called a synaptic end bulb. And look what we see a lot of. There's lots of mitochondria here because nerves are always working, so they need lots of energy. We also see a lot of these round vesicles. These vesicles are synaptic vesicles that once again contain neurotransmitters, and at the neuromuscular junction, it is referred to as acetylcholine, ACH. ACH is always excitatory at the neuromuscular junction. So we have a bunch of these vesicles containing the neurotransmitter. And then we have this space or this gap or this cleft. It's called a synaptic cleft. And all the synaptic cleft is a gap or space between the end of the axon terminal where that end bulb is, and the muscle, which is the motor end plate. So hopefully you're getting comfortable with some of these terms. Axon, synaptic end bulb, neurotransmitters, acetylcholine, synaptic cleft, the muscle being the motor end plate. Now, we're going to take this part of it, we're going to take this square, this box, we're going to blow it up right here, so we're zooming in right here. When we zoom in, we're going to be looking at this part of the synaptic end bulb, and we're looking at this part of the motor end plate. So here is the synaptic end bulb, the very end of it. Here's the synaptic cleft, which would be here. And then here's the pink, the motor end plate, which is here. Now, one other thing I want to point out to you, if we look up on the upper right, is that we see that calcium is influxing. calcium is influxing. It is flowing inward. And this calcium that's flowing in has a very important job. As calcium flows in, it tells these synaptic vesicles that contain a neurotransmitter called acetylcholine to move towards the end of the synaptic end bulb and to open up and release its contents. So we see calcium here coming in, stimulating and acting upon the neurotransmitter vesicle that contains acetylcholine. When it binds to this, you could see that it opens up. And this method of releasing its contents is what we call exocytosis. Exocytosis. Exocytosis is the method of which a vessel opens up and releases its contents outside of it. So calcium came in, you see the acetylcholine is released from the synaptic end bulb, and now it is floating in the synaptic cleft. As it's floating in the synaptic cleft, it needs to bind somewhere on the motor end plate. Well, where is acetylcholine going to bind? The acetylcholine is going to bind to an acetylcholine receptor. It just so happens that these receptors are channels, like a doorway, and it's closed. And when acetylcholine binds to this acetylcholine receptor, which happens to be a sodium channel, it's like open sesame. So you see it binds, the sodium channel opens, and sodium, Na, sodium rushes in. Now, when sodium rushes into a muscle cell, we use some terms like depolarization or action potential or nerve impulse. So from our perspective, the term action potential that we see here, you see electrical activity, so we're going to think there's a nerve impulse. Okay, so when sodium rushes in, an action potential is produced, and that's going to create some sort of excitation to the muscle. Okay, we're going to review that a few more times. So here's just a type of view of the neuromuscular junction and what it looks like. So here's a somatic motor neuron. We could see that we have collaterals. We can see that we have an axon terminal, which is the very end, and way, way at the end of that is the synaptic end bulb. Here is the striated skeletal muscle. So this is what neuromuscular junction really looks like. Now, if this nerve or these axons are damaged or cut, then there's no life force that's making it to the muscle, and a muscle atrophies. It wastes away. It shrinks in size. That's called atrophy. When you have lots of impulses going to the muscle, let's say you're exercising, then the muscle cells start to thicken and grow. We call that hypertrophy. Hypertrophy and atrophy are opposites. So let's take a look at some of these terms, because this is important to know. So what we're looking at here, all of this is part of a sarcomere. A sarcomere is the functional unit of muscle. The sarcomere is the functional unit of muscle. So this portion here, all these small muscle fibers or myofibrils, and we can see deep in here like dark and light filaments, they do look striated. These are going to be your actin and myosin filaments. These are your contractile filaments. You see some mitochondria, lots of mitochondria here because muscles are contracting. It takes energy, it takes ATP. Here is a T tubule. The T stands for transverse tubule. This is where the action potential is going to be traveling. That action potential of excitation that we saw here, that action potential, this right here, this invagination, if you would, is a transverse tubule. It's a T-tubule. It's transverse tubule, this invagination. That invagination is this here. So the impulse is going to travel down this transverse tubule and relay to the blue mesh-like structure, the bluish mesh-like network of fibers here. That's called the SR, or the sarcoplasmic reticulum. The SR, or the sarcoplasmic reticulum, is really important because it stores a very, very important mineral called calcium. Calcium is stored in the sarcoplasmic reticulum. This is going to be an important part of the story when we talk about muscle physiologies. We do hear that calcium is involved in making muscles contract. So let's make sure we understand. T-tubule, you'll see that the sarcoplasmic reticulum kind of butts up against the transverse tubule, and the sarcoplasmic reticulum kind of articulate with one another. Okay, so let's take a look at some of the sequence of events here that takes place. So we have on the top right, well, let's look on the top left. Here's a somatic motor neuron. Here are the dendrites. This is the cell body. Here is the axon. It is myelinated by these Schwann cells. This is the insulation to it. It speeds up conduction of impulses to the very end. You see all these axon collaterals, and at the very end are synaptic end bulbs. Okay, so now the synaptic end bulb articulates with the end plate of the muscle. That is the neuromuscular junction. So here is the nerve. Here is the end plate. These little circles would be the neurotransmitter acetylcholine. So somewhere along the way, calcium came in, told this synaptic end bulb, where these vesicles are, these neurotransmitters, that's the synaptic vessel holding acetylcholine, to release the acetylcholine in the synaptic cleft. So it binds to the synaptic, it travels in the synaptic cleft and binds to the acetylcholine receptor. These acetylcholine receptors are lined all throughout the motor end plate, and that binding of acetylcholine to that acetylcholine receptor, remember we said it's a sodium channel. So the sodium channel opens up, sodium rushes in and creates activation or excitation. Goes right down the T-tubule, the transverse tubule. Now, what did the transverse tubule butt up against? What does the transverse tubule butt up against? The sarcoplasmic reticulum. So what does it butt up against? The sarcoplasmic reticulum. What does the sarcoplasmic reticulum house? What does this store? This blue mesh-like stores what mineral? It stores calcium. So now calcium is released. And as calcium is released, it's going to have some major influence over this sarcomere, where you have thin and thick filaments. These thin filaments are called actin, and the thicker filaments are called myosin. What's going to happen when calcium is released is going to be something pretty magical that happens that I'll talk about shortly, that will eventually make the muscle contract due to the tension that is produced. Okay, so I'm going to take a pause here, and when I come back in the next video, I will get into the different types of muscle proteins.